Today we're looking at the lymphatic organs and tissues, and they're broadly separated into two categories: primary or secondary lymphatic organs. Primary lymphatic organs are the places where the cells, the workers, the soldiers of the immune system are produced. Primary lymphatic organs produce immune cells, and it's where they become what we call immunocompetent. So immunocompetent simply means a differentiated, targeted immune cell that can respond to an invader, that can carry out an immune response. It has its job, and it's programmed. It is immunocompetent. They are produced in red bone marrow or in the thymus. They're all really produced in red bone marrow. T cells start as pre-T cells in bone marrow, and they get their last little bit of programming in the thymus. So we still consider the thymus a primary lymphatic organ, but both B cells and T cells are produced in the red marrow of long bones. Produced in red marrow from pluripotent stem cells, which maybe you know, maybe you don't know, simply means cells that have multiple possible fates. Pluripotent, many possible paths through which they can differentiate. After they've differentiated into immune cells, they're fixed. They don't unprogram and choose something else. They are immunocompetent and have their instructions moving forward. Now, the instructions are not even the focus of this lecture. They will be the focus of Tuesday's lecture when we look at the steps taken in an immune response. So stay tuned for that. Red bone marrow and the thymus produce these soldiers of the immune system, B cells and T cells. So we're not going to look at the process by which they're made. We will assume they are. We want to know how they work and how they react, not necessarily the signals that commit a stem cell to a certain path. The thymus, located here, anterior to the aorta and the great vessels in the mediastinum, between the two lungs, just superior to the level of the heart. It is a gland. It's a pretty non-specific gland. It stains somewhat darkly, and we'll look on the next slide. There's a few internal features to look at, but at first glance, from the outside, superficially, just a general fatty mass that can release immune cells into circulation if needed. But inside, we see a bit more detail. Inside, we see where the T cells are committed to their fates. And I don't want to spend a lot of time on the detail here either. The immune response is what I want to focus on largely, but you can see the compartments within the thymus. And this is a theme for most of the lymphatic organs. The trabeculae, one of which you see at the bottom on the left-hand side, folds in the tissue of the thymus, break it up into little sections or little compartments. Trabeculae break up the organs into compartments. And this is something we'll come back to a few times, so not to worry if it passes you over now. And then within each compartment, you generally have, like in all other tissues, you have a cortex around the outside. Here it's large. This really darkly staining area is the cortex. And the light pink nucleus in the center is the medulla. And as those pre-T cells arrive, they filter in through the trabeculae and they move into the center of that compartment. So they start in the cortex. They are associated with one or two chaperone cells. They are educated in the cortex. And through that process, migrate inwards. And not many of them reach the medulla where they are fully committed and mature T cells. Only 2% of all the T cells that arrive at the thymus differentiate properly, get the right series of marching orders, and then end up as mature T cells. 98% either don't differentiate properly or are discarded. And in that case, they'll be broken down by macrophages. They'll be destroyed, they'll be consumed, their parts will be recycled to be used later on down the road. So the thymus serves as a last central repository for T cells before they're sent off to the rest of the body. Now, after leaving the medulla and then entering into circulation, they will colonize other sites. They'll colonize other organs in the lymphatic system. And it's at these other locations that they will have an immune response, that they'll do their work, whatever that happens to be. We don't know what that is yet. But we will look today at lymph nodes and the spleen and lymph nodules. All of those are sites of the immune response where action is taken against invading microbes or pathogens. So primary lymphatic organs, red marrow of long bones and the thymus, here completed immunocompetent immune cells are produced. And now they venture outwards to secondary lymphatic organs. And the secondary lymphatic organs are what were mentioned on the last slide as the places that the T cells will eventually migrate to and colonize. Lymph nodes. The spleen is a giant lymph node, essentially. And lymph nodules, which a number of them make up each individual lymph node, but they can be found throughout the body as well. And we'll get to that in turn. So the secondary sites don't produce new immune cells. The immune cells that are there can divide and they can take on slightly different roles, but no new ones are produced. If they are taking on new roles, it's part of the immune response. The secondary organs are where the immune response is carried out. And we call them all organs. We call the nodes and the spleen organs because there's a capsule that wraps around that mass of cells. Technically, the nodules, the smallest of these three, aren't organs because there's no capsule. It doesn't really change their function, but just be aware of that distinction as we move through the descriptions. Secondary lymphatic organs, where the immune response is carried out. The most common of these organs are the lymph nodes. One of which I'm showing you here, and we will walk through in a fair amount of detail. And you've probably heard of lymph nodes before. Maybe you've had a swollen lymph node. You can feel some of them in your throat sometimes. There's large masses of them in the armpits or the groin, where there are fatty deposits. Lymph nodes are small bean-shaped filters in the lymphatic system. Let's start with the general structure. We already pointed out part of the general structure when we introduced the thymus. Here again we have trabeculae that are folds in the outside surface of the tissue that create compartments or sections within the organ. So trabeculae, you can see it over on the right-hand side, create compartments. And then with the other structural components, and the other structural components are reticular fibers, they're also listed here, and the capsule itself wrapping around. they create what's called the stroma, the supportive scaffolding onto which the functional tissue is strapped. So the trabeculae don't do anything, the reticular fibers don't do anything as far as the immune response is concerned. They are structural. And that structural portion is called the stroma. Everything else, all the other cells that have a function, an immune function, are part of the parenchyma. Just a broad distinction between the two. So these folds inwards and this fibrous reticular section are supportive, and all the cells in the middle are functional, part of the parenchyma. Now we can break down the tissue within a lymph node into three general categories, three general regions. Again, there's a cortex and a medulla, but we have an added layer of complexity here because there are two portions to the cortex. There's an outer and an inner cortex. And they're generally divided as follows. This is the best representation that I could manage. The two bluish layers are the cortex, and the red layer near the center is the medulla. The outer cortex is this light blue, the largest band around the outside. And it is characterized by all of these darker staining, smaller nodules. And these darker staining little bubbles in the outer cortex are lymphatic nodules. So lymphatic nodules, we've seen them in the list of secondary lymphatic organs already. Here they are inside a lymph node. They can also exist outside of the lymph node in other connective tissue. But it's the same structure. It's a lymphatic nodule. Here it's inside the outer cortex of the lymph node. These are important because this is the first site of the immune response, of the adaptive immune response, where B cells and T cells recognize an invading pathogen, and then they differentiate and execute a response. We don't know about the mechanics of that yet, but we'll get into it. And when we do, we are essentially residing within one of these lymphatic nodules. That's where we'll spend a lot of time there. Now there is more detail if you want to look it up in the book, and I thought, let's not put so much detail in. I don't even know if it's necessary to know that the nodules change in shape, but it illustrates how a lymph node can swell and grow in size in response to an invading pathogen. All of these little dark staining circles start as really small primary lymphatic nodules. And when they need to respond to an invading pathogen, they grow and the cells inside divide. They become a secondary lymphatic nodule. So, if that's the outer cortex, this darker blue area is the inner cortex, and it is devoid of lymphatic nodules. It has some other cells, but none of the locuses where the immune response is carried out. And then the medulla in the center, still another variety of cells, and it's the outlet for lymphatic flow after the incoming fluid has been filtered by the lymph node. So the meat and potatoes of the action in the lymph node is done in these lymphatic nodules. You have all the characteristic cells that you need in order to mount an immune response. B cells, we'll see what these dendritic cells have to do. They grab and position microbes and allow the B cells to do their work. There are a ring of B cells around the outside of each nodule, and it's sort of like some B cells will specifically recognize a certain microbe or pathogen. Others don't. And the others will move to the outside so as not to get in the way. The B cells that aren't active or involved in this immune response push their way to the outside of the lymphatic nodule. In the medulla, T cells are mostly present, also some dendritic cells to help position the microbe or the pathogen if a T cell response is required. And again, we don't know what those responses are yet, before eventually reaching the medulla, where we have outgoing B cells, plasma cells that are activated B cells, and other things that need to circulate through the lymph to other parts of the lymphatic system or be returned to the blood. This is the outlet for flow through the lymph node. And speaking of which, it makes sense to focus on flow through the lymph node. As a filter, fluid or lymph is pushed through from the outside towards the center, from afferent lymphatic ducts, or vessels rather. Afferent arrives at a certain structure, efferent leaves. Notice the afferent vessels with their one-way valves. There's many of them on the left-hand side of this figure. And this detail that I've added is slightly different than what's included in the figure in the book or in the slides if you're looking at them now. I don't care necessarily that you know the names of all the sinuses, of all the passageways, just that they proceed from the outer cortex into passages through the inner cortex, through the medulla, and then eventually out the efferent lymphatic vessels. Note the efferent lymphatic vessels also have one-way valves, so no backflow can occur, and they emerge very similarly to the kidneys at the hilum. In fact, these look like small kidneys with a couple different afferent inputs. So these are tiny bean-shaped filters. There are hundreds to thousands of them scattered throughout the lymphatic system. Some larger invading pathogens can be trapped outside for macrophages to break down, but the large majority of the immune responses we'll look at are done in those secondary lymphatic nodules. So those are pretty important. The cells in that region are important. The outer cortex is important. All the structures are important, but we're going to spend more time revisiting the lymphatic nodules. You can kind of see what that looks like in practice here. I didn't include a lot of detail, and I tried to highlight the lymphatic nodule for you. It's really faint in the picture in the book. It's hard to discern that there is any variation in the tissue. It's fairly large here, and it's comprised of all the cells that we've talked about. The node itself doesn't necessarily look like it was shown in the cartoon. It kind of looks like a cancer cell reaching out with all its projections to find other cells to trap or places to divide into. But that's a lymph node in practice with the macrophages and the lymphocytes inside. So more good detail coming on what happens inside the nodule. But this is the setup. Any invading pathogen, anything through the lymph has to pass through this location first. So all the immune cells get a good crack at it. The spleen is not very different, to be honest. The organization of structures within the spleen, although named differently, follow a very similar hierarchy and flow. Very similar overall structure as well. The main difference is that there aren't lymphatic nodules that elicit an immune response. Here instead flow through the spleen that arrives from the splenic artery and returns out through the splenic vein, not lymphatics. Flow through the spleen occurs through these different pulp regions. There are two different regions that are referred to as white pulp and red pulp. And white pulp is outlined here, the white area bordering the central artery as it enters the branch of the spleen. And red pulp is the area in between. On the figure on the right-hand side, it almost looks backwards. The white pulp stains darker than the red pulp. But the spleen also carries out an immune response to any pathogens that are in the blood proper. In the white pulp, where arterial blood is first delivered, B cells and T cells are present. A lot like the lymphatic nodules in the lymph nodes, B cells and T cells are present. And so, if anything comes into the spleen that is recognized as a pathogen, or microbes come into the spleen carried by the blood, the B cells and T cells in the white pulp will recognize it and carry out an immune response. Macrophages and or neutrophils will carry out phagocytosis to break down any constituents that can't be destroyed through the immune response, all in the white pulp. So blood flows in through the white pulp, following this black arrow on the screen, out through the red pulp and drained eventually through the splenic vein. Now the red pulp has three main roles, unrelated to the immune response, and really one primary role in adults. 1.5, maybe 2. The red pulp contains enzymes that deal with any old, worn out, or non-functional red blood cells. So the spleen is the site of red blood cell recycling, or the major site of red blood cell recycling in the body. We start to break down erythrocytes in the spleen. And we won't look at the details involved in that process, but the red pulp area of the spleen is where those steps are taken to break down and recycle components of the red blood cells. It's also tasked with storing platelets, and we haven't looked at platelets in depth. They are one of the formed elements in blood, along with white blood cells. Platelets are primarily involved in the clotting cascade, but we're not concerned with their role as far as the immune system is concerned, so we're not discussing them at length. And lastly, in fetal circulation, the red pulp is responsible for producing fetal hemoglobin and fetal red blood cells. That stops after birth, and red blood cells are produced in the red marrow of long bones in adults. So that's not a major function of the spleen in adults, obviously. So you can see a very similar organization. Same kind of structure, different compartments. Overall, the bean shape with a hilum is obvious. The B and T cells get first crack at any incoming fluid, here the blood, in case there are microbes in the blood, to mount an immune response, and then act accordingly. So the spleen, very similar in schematic to the lymph nodes. And it's worth bringing up the lymphatic nodules here, even though we've covered them in a fair amount of depth already. They're the same structures that we saw in the lymph nodes proper. These are masses of lymphatic tissue that don't have a capsule surrounding them, and they can be scattered throughout the mucous membranes and connective tissue of various tissues in the body. Wherever those masses happen to be, you would find lymphatic nodules. We just identified ones specifically that were in lymph nodes in our discussion earlier. A way that we would differentiate these from the nodules within a lymph node is that these are often referred to as mucosa-associated lymphatic tissue, or MALT. A neat acronym that you can use to differentiate these from the ones in the lymphatic nodes, or the lymph nodes. And often you'll see these aggregate together in places like the appendix. You have these fatty masses that aren't lymph nodes themselves. They're aggregations of lymphatic nodules, or the tonsils are an example of an aggregation of lymphatic nodules. They'd be able to carry out the same immune responses as the lymphatic nodules in the lymph nodes, but the directed flow of lymph through those tissues doesn't happen in as ordered a manner, let's put it that way. So same idea, same cells present in order to mount an immune response, and we'll learn the details of that response next week. They are just not encapsulated, and they're spread out throughout connective tissue in various other tissues of the body. A very similar role and structure. So that's a high-level overview of the tissues. And we know the flow of lymph throughout the system. We know the flow of lymph through the individual tissues. And I think now we can talk about the specifics related to immunity. And if we just cover innate immunity today, we'll be on pace, which is good, to get adaptive immunity, which is, I think, the most important part of this section next week. But innate immunity is broad and non-specific. It is present within us, and it's always present within us. It describes a host of responses that are generalized. They respond to a number of different pathogens, a number of different released signals from those pathogens, toxins, metabolites, etc. And we've always been, this has always been present, and it always will be present as long as we live. It's an innate characteristic of our biology. So these are physical characteristics. Physical characteristics. We can break up the innate immune system into two main branches, or two main components. The external or internal defenses, the external barriers, or the internal enzymes or proteins that degrade a broad spectrum of invaders. And the external barriers are things like the skin and mucous membranes. The internal defenses are some leukocytes, some white blood cells, and other proteins that respond in a very general and broad way. They'll always respond to a wide variety of abnormal tissues or pathogens. So innate immunity, skin and mucous membranes, the external first line of defense, is mechanical. The skin provides a really nice natural barrier. Way back in section 1 of semester 1, when we looked at the integumentary system, we saw the packed and ordered layers of keratinized skin cells. They create a really nice, definite barrier against invading microbes. And then the fact that we tend to lose those skin cells. We shed skin cells periodically. That helps to remove microbes from the surface of the skin. So the skin, mucous membranes. Mucus is a viscous, thick, sticky fluid that will trap some, but not all, invading microbes. And then depending on where that mucus is produced, you can either expedite those microbes out of the system by sneezing or coughing, helping to expel some microbes, Or maybe they're washed downwards towards the stomach, where the gastric juice and its really high acidity will destroy most of those invading microbes. Here we have physical defenses, the skin, the mucus, the cilia that move that mucus and that fluid. Urine flow, vomiting, defecation, those are all processes that move substances through the body. And then through expelling those substances, we also expel microbes. All help to protect the body from invading pathogens. To complement the physical defenses, the mechanical defenses, we're already aware of some broad chemical defenses, enzymatic defenses, metabolic defenses. We've seen lysozyme at least twice. A broad spectrum antibacterial agent. It's in saliva. We've talked about that. We haven't talked about it being in sweat or tears or nasal secretions, but it is. And it will break down invading bacteria in a general sense. Most invading bacteria in a general sense. Gastric juice is a really high acidity, a really low pH to break down microbes that enter the stomach. And then sebum, which is that oily substance secreted onto the surface of the skin. Sebum contains some unsaturated fatty acids that inhibit fungal growth. So chemical prevention of the spread of pathogens in the body. These differ from the internal defenses. If microbes ever pass this layer, this first line of defense, they come into contact with, or they elicit reactions from, the internal defense mechanisms. There are three of them. Antimicrobial proteins, we have phagocytosis, we have inflammation and fever. We'll talk about each of those in turn. They do invoke some chemical pathways, but they're separate from the first line of defense that we just talked about. So there are four main kinds of antimicrobial proteins. And antimicrobial proteins, like most proteins, will typically execute or facilitate a reaction. They'll do something to the invading microbes. Antimicrobial proteins can be signals to the immune system. And interferons are an excellent example of that. Interferons are small sequences of proteins that are released when a cell gets infected. When microbes invade a healthy body cell and it becomes diseased, or when a virus invades. When that happens, that healthy cell, now diseased, will secrete or produce what are called interferons. And interferons, they don't interfere like their name would suggest directly, but they recruit B cells and T cells and other cells of the immune response to say, hey, I'm infected, you have to shoot the messenger. Otherwise, the body is at risk. So the interferons recruit other cells to break down and destroy the infected cell. We'll see a bit of the complement system. These are a series of proteins that are like interferons. They're produced to magnify the immune response. When a cell has been infected, we want the immune response, and we want the immune response to be boosted or bolstered. We want a large immune response to make sure that the infection is stopped. So the complement system, or complement proteins, will boost the normal immune response. And we'll see that coming up next week. Iron-binding antimicrobial proteins simply steal iron away from incoming bacteria. Iron is often used by incoming bacteria to carry out chemical reactions. And so if it's not available, they can't carry out those reactions. So iron-binding proteins are those that they'll chelate, they'll bind, they'll store, they'll keep away iron from invading bacteria. And lastly, but not least, we have proteins like the ones that I talked about last class briefly. They're named defensins. They are the tiny protein spears that will poke holes into bacterial membranes, causing them to burst. Some of them will help to recruit immune cells to the site of an invasion. They're physical fighters in the immune response. Defensins will poke holes directly into a bacteria to make it burst, to release whatever the contents are, and then the contents can be destroyed by the next internal defense mechanism. So if a cell is damaged, these proteins might come and poke holes in the cell. It's going to destroy the cell, so it's not going to continue to function. The microbes that originally infected it might be released. They're not destroyed yet, but now they are freely available for phagocytosis to break them down. So there are various phagocytes that will carry out phagocytosis, and natural killer cells, which we saw last class, that are general killer cells, general B and T cells that are non-specific and will destroy a wide variety of abnormal cells within the body. So natural killer cells are part of the innate immune system. They are the lymphocyte that is part of the innate immune system. B cells and T cells are part of the adaptive immune system, immune response. That's coming up, but I want to make that distinction now before we move forward. Now, cells in the body are easily recognized by the immune system. They have a fingerprint. The proteins in the membranes of our cells have a certain fingerprint that we recognize or don't recognize. And natural killer cells, if they don't recognize the fingerprint, will attack and kill that cell. Usually it's abnormal, usually it's an invader. Sometimes it's a transplant, but it still has that abnormal fingerprint. And natural killer cells will try to do that to tumor cells, to cells that have been virally infected, etc. They will either institute a chemical reaction within the cell that causes it to stop working and eventually implode. They can make the cell rupture and burst. The mechanisms by which we will look at in depth, they're part of the immune response in the next section. But they can make holes in the membranes, and then other proteins can dive in and wreak havoc inside the cell. They don't kill the microbes, but the microbes are then destroyed by the phagocytes that are in the immediate surroundings. They will consume and then degrade any microbes that are released by this rupture, by the apoptosis of cells. That's a fun word. Apoptosis simply means programmed cell death. Every cell in your body will eventually undergo apoptosis. Natural killer cells might accelerate that process, turn that program on before it's time to kill the cell and the invading bacteria. So natural killer cells will open up the infected cell for degradation. The microbes then will be degraded by phagocytes such as neutrophils and macrophages. Those are our main phagocytes in the body. Remember, macrophages differentiate from monocytes. Monocytes turn into macrophages. Those are one of the kinds of white blood cells we saw at the start of the section. Those two cells are responsible for, just like this image is showing on the right-hand side, eating the microbes. Put a pair of googly eyes on this thing and it's eating the microbes. What does that process look like? You've seen phagocytosis before, but just a brief overview. 5 steps, which I'm not going to go through in detail, but I do want to point out at least 2 of them. And the first is an important one, chemotaxis, which I referenced briefly in a previous class, is the first signal to initiate phagocytosis, to consume and degrade those microbes. Whatever the signal is, chemotaxis describes the broad umbrella of signals. Chemotaxis is the signal. This is also the thing that initiates emigration of white blood cells from the blood. Remember we talked about white blood cells rolling along the endothelial cells, and they have those proteins that will eventually lock up, and they'll squeeze between the endothelial cells when an immune response is required. How do they know to emigrate at one location? They emigrate because chemotaxis is a signal that says we need an immune response here. And one of the downstream effects is to synthesize those proteins that trap the white blood cells as they're moving through the body. So chemotaxis initiates the series of steps that allow white blood cells to emigrate. It also initiates the signals that prompt the phagocytes to engulf and break down the microbes. The phagocyte will surround it, it will consume it, it will bring it in by endocytosis. You have the microbe within a vacuole here that eventually will merge with a lysosome. Conveniently enough, lysosome contains lysozyme, the candidate or gold standard broad spectrum antibacterial enzyme that we've talked about now 4 or 5 times. And it injects lysozyme into that little bubble so that it can degrade the microbes in an isolated and intact environment. Targeted degradation of that microbe to remove it from the circulation, to protect the rest of the cells from its activities. Most things will be broken down and recycled and used. Anything that's not is left in residual bodies that will be expelled, discarded. So this is one mechanism by which we break down those microbes. Lysozyme is one standard route to break down microbes. They also use oxidant bursts. You've heard of antioxidants, right? Antioxidants are good for you to protect against oxidation. Orange juice has lots of antioxidants in it. You want antioxidants to make you live longer. We don't want microbes to live longer, and so we do the opposite. We bombard them with oxidants, free radicals, reactive oxygen species, superoxide. You'll hear about these things in upper level classes. But we bombard them with detergent-like or really abrasive signals that will cause them to break down, as well as bathing them in lysozyme. Now, I'll throw up a couple last points on fever and inflammation, so you can frantically copy them down in the last 3 minutes. This is kind of a side note. I don't really care to go in depth about the sequence of events or the cascade that initiates the fever response or the inflammation response. It is large It is vast, and it's the topic of whole courses, so we're not going to look at the inflammatory response, NF-kappa B, and all of those intermediate signals. But it's worth pointing out that inflammation is a non-specific set of responses that accompany the immune response. And those responses are an increased blood delivery to the site of the immune response, an increased local temperature at the site of the immune response, increased, I'm losing the word. What's the word for porousness? Porosity? No. Spaces between the endothelial cells. It opens up the space between the endothelial cells, allowing things to emigrate more easily. I'll use a large description of it rather. I know I'm going to remember it as soon as I finish up lecture. All of those things act to get more blood to the area, get more immune cells to the area, and speed up the activity of those immune cells. Faster temperature, everything happens faster. All reactions happen faster. That constitutes inflammation. When that happens at the whole body level, it's called a fever. And that's really all I care to talk about, inflammation and fever. There's some pretty interesting stuff that we can get into, but we're not going to. We want to focus on the immune response. That's coming up next. Have a wonderful weekend.